Have you ever just stopped and looked at a towering oak tree, or, your pet dog, and actually asked yourself what they're made of at the absolute fundamental level? I mean, beyond the organs, well beyond the cells. When you zoom right down to the bare chemical foundation, what are the physical building blocks that actually make life possible?
Okay, let's dive into this. Think of life not just as some abstract biological process, but as the ultimate grand building project. In this explainer, we're basically going to read through the user manual for your own biological machinery. We'll be looking at the massive, complex structures that form the absolute foundation of literally every living thing on Earth. Now, to understand this architecture, we really need to know our materials, right? A macromolecule is exactly what it sounds like, a massive, highly complex structure. But nature doesn't just spontaneously generate these things out of thin air. It uses a brilliant modular system of what we call monomers. Think of monomers as individual repeating subunits, like tiny Lego bricks. On their own, they're relatively simple, but when you snap them together, when they're polymerized by the thousands, they build the staggering complexity of a macromolecule.
So how do your cells actually forge these biological subunits together? Well, it relies on an incredibly neat trick called a dehydration or condensation reaction. And honestly, the logic is right there in the name. To join two small monomers together, the cell literally pulls a molecule of water out of them. By removing that H2O, a strong covalent bond is forged, locking the bricks together. It's basically the biological glue that builds life.
It turns out all of the staggering, infinite cellular complexity in your body boils down to just 4 main categories of macromolecules. We've got carbohydrates, lipids, proteins, and nucleic acids. And that right there is our roadmap for today. Let's kick things off with our first pillar, carbohydrates, which are all about energy and structure.
Chemically speaking, carbohydrates are molecules made mostly of carbon, hydrogen, and oxygen. They're essentially biology's primary fuel source. And this brilliantly illustrates how nature uses these structures across totally different forms of life for 2 very distinct functions: storing energy and building structures. For energy, plants link simple sugars like glucose into starch, which they stash away in seeds and potatoes. We animals, on the other hand, link our glucose into a much more highly branched chain called glycogen. We stockpile that in our liver and muscles so we have a quick energy boost ready to go. But remember, carbs aren't just fuel, they're serious building materials. Plants build their cell walls with straight, alternating chains of glucose called cellulose, which is exactly what we call dietary fiber. And this is wild. Animals and fungi actually modify their glucose with nitrogen to make something called chitin. Chitin is the exact stuff that makes up the crunchy exoskeletons of insects and crabs. And get this, a fascinating little fact, it's also used to make surgical thread that naturally dissolves inside your body over time. How cool is that?
Moving right along to pillar number 2, lipids, or, as I like to call them, the water haters.
Now, lipids are a super diverse group, but they absolutely all share one universal trait. They are hydrophobic. They actively repel water, basically acting as biology's ultimate waterproofing material. And this chemistry directly connects to your daily life, especially your diet. Fats or triglycerides are a type of lipid made of a single glycerol molecule attached to 3 long fatty acid tails. But here's the kicker. The physical shape of those tails is what dictates your health. Saturated fats only have single covalent bonds. This makes their tails perfectly straight, allowing them to pack really tightly together. That's why things like butter and lard are completely solid at room temperature, and unfortunately, it's also why they can collect in your blood vessels and cause heart disease. Cis unsaturated fats, however, have double bonds, and those double bonds create a literal kink or bend in the tail. Because of that kink, they can't pack tightly together, so they stay liquid at room temperature, like olive oil. These are much kinder to your heart, but you definitely want to watch out for trans unsaturated fats. These are artificially manipulated to remove those kinks, making them solid again and highly, highly unhealthy.
But hey, lipids aren't just about storing fat. They also act as the ultimate cellular bouncers. The phospholipid bilayer is incredibly unique. It has a water-loving or hydrophilic head and 2 water-hating, hydrophobic tails. When you drop these into an environment with water, they naturally, automatically arrange themselves into a double layer. The heads point outward toward the water, and the tails hide safely on the inside. This creates a really strong, flexible membrane around absolutely every single cell in your body. It's actually very much like the flexible, delicate membrane that holds an egg yolk together. Before we wrap up lipids, we should definitely clear up a really common misunderstanding about steroids. When we hear the word steroids, we usually think of, athletes getting into trouble. But biologically speaking, steroids are just a normal class of lipids defined by a specific structure of 4 interconnected rings.
Take cholesterol, for example. It's a steroid, and it has a totally vital, natural job, keeping your cell membranes beautifully flexible. We literally make it right in our livers. Other steroids act as essential hormones, things like testosterone, estrogen, or even cortisone, which helps treat allergic reactions by directing very specialized cellular tasks.
Okay, let's look at our third pillar, proteins, the cellular workers.
So the crucial point here is that if carbohydrates provide the fuel and lipids form the walls, proteins are the dynamic, hard-working machines doing the really complex work inside the cell. They act as enzymes, hormones, membrane channels, receptors, you name it. And here is a completely staggering number to consider: 20. Absolutely every single wildly different complex protein in your entire body is built from a menu of just 20 possible ingredients. We call these ingredients amino acids. Using that exact same dehydration reaction we talked about earlier, your cells just snap amino acids together with strong peptide bonds to create these massive long polypeptide chains.
But as you might guess, a long floppy string of amino acids doesn't actually do much on its own. To become a working machine, it has to fold. And we can trace this structural evolution through 4 distinct levels. First up is the primary structure. This is simply the exact linear sequence of amino acids, which is determined directly by your DNA. Next is the secondary structure. This is where the chain locally folds into a twisting helix or a pleated sheet, mostly held together by little hydrogen bonds. Then we get to the tertiary structure. This is where the entire chain folds in on itself into its final, distinct 3D shape, becoming a fully functional, globular, or fibrous protein. And sometimes life takes it one step further to a quaternary structure. This happens when 2 or more separate proteins link up together into a massive working complex. For instance, hemoglobin, the crucial oxygen carrier flowing through your blood right this second, is actually a massive quaternary complex of 4 individual globin proteins working together in perfect unison. Finally, we arrive at our fourth and final pillar, nucleic acids, the ultimate blueprints of life.
If proteins are the workers, how on earth do they know what specific sequence of amino acids to form? Well, that's the job of nucleic acids, the macromolecules that actually store and transmit all your inherited information. These biological instruction manuals are built from their own specific monomer bricks, called nucleotides. Each nucleotide has 3 distinct parts: a monosaccharide sugar, at least 1 phosphate group, and a nitrogenous base. Now, these bases come in 2 flavors: single-ring structures called pyrimidines and double-ring structures called purines. The cells link them all up so that alternating sugars and phosphates bring the rigid, incredibly strong backbone for our genetics. Now, what's really interesting about this slide is when we put our 2 great nucleic acids head-to-head, some incredibly distinct differences emerge. DNA, which sits safely locked inside the nucleus of our cells, is the permanent archive. It uses the sugar deoxyribose, it forms a double-stranded anti-parallel helix, and it uses the base thymine. RNA, on the other hand, is the smaller, temporary working copy. It's single-stranded, uses ribose sugar instead, and actually swaps out thymine for a base called uracil. But the true, undeniable magic of these blueprints lies in the strict hydrogen-base pairing rules. Adenine always pairs with thymine or uracil in RNA, and guanine always pairs with cytosine. That's it. This amazingly simple, elegant matching rule is exactly how all genetic information is accurately copied and passed on from one generation to the next.
Which perfectly brings us back to where we started, but now with a completely new, profound perspective. You, me, the plants right outside your window, the microscopic bugs in the soil, we are all built from carbohydrates, lipids, proteins, and nucleic acids. So I want to leave you with this final thought-provoking question. How do just 4 simple categories of molecules combine, fold, and bond to create the staggering, infinite diversity of all life on Earth? It's the ultimate puzzle, isn't it? And understanding these macromolecules is literally just the first page of your own biological user manual. Keep exploring.